So your best friend came to you with concerns regarding their home test kit. It said they are predisposed to type 2 diabetes, they don't eat sugary foods, and they want to know more about this perceived risk. Let's help them process and explain why this might be less scary than they perceive. Let's talk about home genetics test kits. When you took this test, you submitted a sample that analyzed your DNA. DNA is comprised of combinations of letters that are paired together in a way that is called gene sequencing. Most of human data is the same, but the area they highlight in your test is not identical and flagged your diabetes risk. Being at increased risk for a disease doesn't mean you will definitely get the disease. There is the potential for multiple genes to trigger this flag, and I'll go over that in a second. First, let's talk about what this specific result can mean for you. When reproduction occurs, each parent contributes genetic data in the form of chromosomes. These chromosomes carry genes that give us our eye color, hair color, and height when expressed. The genes break down further into alleles, or data from each parent. The pairing of the parent's alleles create the trait that is expressed. An example of this is hair color. The mother's data may be an allele that expresses brown hair, while the father's allele may be blonde. The combination may result in a lighter shade of brown hair when the gene expression occurs with you. The reason your hair is brown is because alleles are considered dominant if they are a more prominent trait, or recessive if it is less influential in gene expression. For most conditions, people have a 1 in 1,000 chance of getting a disease, but you have a marker that triples your risk. That means your risk is 3 in 1,000, which still isn't very high. These markers indicate an increased risk for a condition, but they aren't accurate predictors of actual risk. In the case of type 2 diabetes, there are other factors to consider. With over 500 variants associated with type 2 diabetes and multiple factors influencing the expression of this gene, type 2 diabetes is considered to be polygenic and multifactorial. Being a polygenic disease, there isn't a single genetic inheritance pathway or guarantee of manifestation if the gene is carried. Expression of this gene can be initiated by environmental factors and multiple genetic contributors, with environmental factors being poor diet, inactivity, and obesity. A gene expression analysis revealed 539 genes, with only 20 being common among those affected by diabetes. A noted environmental factor of system dysregulation, such as obesity or circadian entrainment, being a factor with the gene expression. Your concern is valid, and it's appropriate to investigate this further to determine your actual risk and modifiable factors. Genetic counselors are experts in the field with the education and resources necessary to monitor and test for conditions with better accuracy than the at-home test kit. Meeting with a counselor and sharing your lifestyle factors, family history, and concern for genetic predisposition gives them the background and information necessary to test and counsel you on your actual risk. The home test kits may also be misconceived as medical grade, clinically valid, and scientifically accurate, which makes following up and having the proper testing and counseling even more important. All that being said, to tell you that just because the gene was found in your study, it doesn't mean you'll necessarily get diabetes. Just like the blonde hair allele from your dad, there are bits of genetic data that you carry that are not expressed and will remain dormant within you. I'd recommend taking your test results and concerns to your primary care provider. Get the referral to the genetic counselor and get the proper testing and have that conversation about necessary lifestyle changes, monitoring, and actual predicted risk.